Hello, my name is Maya Pulliam, and today I'll be talking to you about the relationship between first-line antiretroviral therapy and blood-brain barrier function. So just as a quick overview, I will be going over HIV, antiretroviral therapy, issues with antiretroviral therapy, and the blood-brain barrier in my introduction. And from then, I'll move into my question, my results, methods, and my conclusion. HIV is a virus that infects immune cells, and as a result, the immune system of those with the virus becomes compromised, and it is more difficult for the bodies of these people to fight off disease and infection. The virus can be treated using a class of drugs called antiretroviral therapies or ART. However, the virus cannot be cured. Antiretroviral therapies consist of multiple drugs that target the virus at various points of replication. Because these drugs are typically given in combination of 2 or 3 drugs, they are very effective in making the virus undetectable in patients. This is very important because that means that 1, the virus is less transmittable from person to person, and 2, those that have the virus have a longer life expectancy. 23.3 million people globally are currently taking ART, which is either to treat HIV that they currently have or to prevent themselves from contracting HIV. 3 first-line antiretroviral therapies I will be focusing on today are the drugs tenofovir, entrectinib, and dolutegravir. Issues with ART. So HIV has been seen to enter the brain as early as 1 week post-infection. As a result, those with HIV tend to have some form of cognitive ailment. Although they are typically mild, these ailments can range as severe as HIV-associated dementia and other issues similar. It is unclear as to why these issues continue to persist even when virus is undetectable. Some antiretrovirals have also been seen to be neurotoxic, and together these 2 details may suggest that antiretroviral therapy is causing its own issues in the brain. This brings us to the blood-brain barrier. This is a protective membrane around the brain that keeps out harmful substances while letting in what the brain needs. The blood-brain barrier is made up of a multitude of cells, but I will be focusing on endothelial cells that line the blood-brain barrier as they express a multitude of nutrient transporters and proteins that help seal the barrier. This includes claudin 5, including GLUT1, which I'll explain further in a minute. But the blood-brain barrier is important because drugs have to get through it to get to the brain to treat virus, so the 2 must interact. This is a model of endothelial cells of the blood-brain barrier. As you can see, they express nutrient transporters, GLUT1, which is on the left, and is held together by multiple proteins, which is seen more clearly on the right. GLUT1 is extremely important as a nutrient transporter, as is the main transporter for glucose at the blood-brain barrier, and glucose is the main fuel for the brain. Likewise, claudin 5 and occludin are very important for helping seal the barrier, as literature has shown that without one of them, the blood-brain barrier has been seen to break down, meaning that the brain is not protected from harmful substances without them. The effects of antiretroviral therapies on people in the body are variable. The drugs that we use today are, of course, considered non-neurotoxic, but even some of these drugs, such as tenofovir and entrectinib, have been seen to cause neuropsychiatric manifestations such as headache and confusion. Literature also suggests that antiretroviral therapies may cause endothelial dysfunction, which leads directly into my question. Does antiretroviral therapies affect nutrient transport to the brain and the integrity of the blood-brain barrier? In order to reduce a bit of the confusion as much as possible, I have split up my methods and results into 3 sections, and that's because I have 3 methods. So going forward, I will explain the method that I use and then directly show the data that goes with that afterwards, instead of explaining all of my methods and then showing all my data at the end. So to start off each of my methods, I cultured primary human brain endothelial cells and then treated them for 24 hours with either tenofovir, entrectinib, or dolutegravir by itself, or with some combination of 2 or all 3 of the drugs. For my first method, I analyzed the total protein of my samples. So to start off, after my cells had been treated, I would lyse them for protein, meaning that I would use chemicals to rupture the cell membrane that didn't be near to the protein that I wanted, and then I separated the protein from the cell part. Then I quantify the amount of protein in each of my samples using a method called Bradford assay, which is in the image to the left. This involves me adding my samples to a plate and adding dye, which turned blue when there was more protein present and more brownish or yellowish when there was less protein. Using the concentrations I got from this, I was able to normalize the amount of each sample I used to conduct a Western blot, which can be seen on the right. The Western blot shows how much of the specific protein I'm looking for is in my samples. So thicker and darker bands indicate more protein and thinner, lighter ones indicate less protein. As you can see, I have two graphs. One indicates the fold change in total protein when the drugs were given individually, and the second indicates fold changes in total protein when the drugs were given in combination. For example, on the left, you can see that emtricitabine and tenofovir, when given individually, cause a fold decrease in claudin 5 protein. And then you can see on the right that when emtricitabine and tenofovir are given in combination, there is a fold decrease as well in total claudin 5 protein. It can be seen that the expression in total claudin 5 protein changes based on the treatment of drugs given. However, none of these changes have been seen to be statistically significant. So looking at changes in total occludin protein, you can also see that there are changes in the expression of occludin based on treatment. However, as I mentioned before, there seems to be no statistical significance in this data set as well. For GLUT1 total protein, like the last two data sets, we see variation in expression of GLUT1 total protein based on the treatments and based on the differences in treatments. However, also like the previous data sets, there's no statistical significance. And it also may be of note that amongst all these three data sets, there seems to be no exact clear trend between the changes in treatment and the resulting changes in fold change. For example, when the drugs are given individually, there seems to be no direct correlation between the resulting fold changes of the drugs when given in combination. So for example, we can see right here that emtricitabine and dolutegravir, when emtricitabine and tenofovir are given individually, we see both cause a slight fold increase. However, when the drugs are given in combination, this fold increase doesn't seem to be additive. So it's not clear if there is an actual trend between the two. So for my second method, I looked at membrane proteins. So unlike my previous method, I did not lyse the cells that I used for this method. I instead fixed them in tubes and added antibodies that fluoresce in the presence of protein I was looking at. And I then, with the aid of my mentor, used flow cytometry to analyze changes in membrane protein. And you can see a bit of what that data looked like on the right. So due to time constraints, I was unable to perform this method on all three proteins. Instead, I was only able to use it to look at GLUT1. And the same can be said for my third method. We can see once again that there are changes in GLUT1 membrane protein based on the treatment. Treatment with any drugs individually result in some type of fold increase in GLUT1 membrane protein. And you may also notice that with treatment of emtricitabine and dolutegravir in combination, there is a noticeable increase in GLUT1 membrane protein. However, once again, this data set has not been seen to be statistically significant. So for my final method, I looked at GLUT1 mRNA. Similar to the first method, I lysed my cells, but this time for RNA, and then separated the RNA from the cell parts. Then I used NanoDrop, which you can see on the far left, to quantify how much RNA I had in each of my samples. And then using that data, I normalized how much of each of my samples I used to convert into cDNA. And the cDNA made my RNA double-stranded, and it acted as a template for my QRT-PCR. I then used PCR to amplify the genes for my protein to examine changes in GLUT1 mRNA. And the mRNA is important because it is what is used to make the GLUT1 protein. So changes in GLUT1 mRNA can be seen based on the different treatments. Most notably, there's a very large increase in GLUT1 mRNA when tenofovir and dolutegravir are given in combination. However, once again, none of these points have been seen to be statistically significant. So in summary, although there is variation in expression of Claudin 5, including GLUT1, based on the differences in treatment, none of the changes have been seen to be statistically significant. So in conclusion, GLUT1 is likely not affected by first-line antiretroviral therapy drugs that I looked at. However, more analysis should be done to examine the effects of these drugs on Claudin 5 and including to determine the full effects that these drugs have on those proteins. Likewise, more proteins and transporters of the blood-brain barrier should be examined going forward because there is a great deal of them, and all of them are important for the integrity of the barrier and brain function. So if any issues exist between interactions with these transporters and proteins and these antiretroviral therapies, then that may indicate the cause of the problem that exists currently. Overall, this data is very important for populations taking antiretroviral therapy, which includes those with and without HIV. We do not want these drugs to be causing unintended side effects on those whose bodies have already been compromised by the virus, and also in those who are currently healthy and trying to prevent themselves from contracting this virus. To end off, I would like to give a special thanks to my mentor, Dr. Deanna Williams, my supervisor, Mrs. Lisa Friedman, and our research coordinator, Dr. Nicole Rosen. Thank you all so much for your help and support throughout my project, as well as your patience with me. None of this would have been possible without you, and I deeply appreciate all of your help and support throughout all of this. Thank you so much.